Okay, let's unpack this. We are looking at what might just be the holy grail of neuroimmunology. It's a medical mystery that has plagued doctors for decades, and honestly, it's one of those things that once you hear it, you can't unhear it.
It really is. It's arguably one of the most stubborn riddles in all of modern medicine.
We're talking about the link between the Epstein-Barr virus, EBV, the virus that causes mono and multiple sclerosis, MS. We all know this statistic, right? The EBV infection is basically a necessary prerequisite for getting MS.
Mm-hmm. If you don't have the virus, you almost certainly don't get the disease.
But that's the thing that's so weird.
Exactly. It's this paradox that drives researchers absolutely crazy. Something like 95% of all adults carry this virus. Almost everyone has EBV. And yet, very, very few people develop MS. So the question for years hasn't been, is there a link? But if the virus is the gun, what actually pulls the trigger?
And today, we think we might have the answer. We are doing a deep dive into a groundbreaking paper from Cell by Kim and colleagues. The title is, get ready for this, Myelin Antigen Capture in the CNS by B cells expressing EBV latent membrane protein 1 leads to demyelinating lesion formation.
It is a mouthful of a title, for sure. But what it describes is a mechanism that's, it's elegant, terrifying, and just incredibly specific. It answers the how.
Right. So I want to set this up like a detective story, because that's really what it feels like. We have our suspect, EBV. We have the victim, myelin, that protective sheath around our nerves.
And the weapon, the autoantigen.
And the weapon. But we've never been able to figure out the opportunity. How does a common virus turn a normal immune cell into, a brain-eating saboteur?
That is the perfect way to frame it. And the protagonist of our story is, maybe surprisingly, the B cell.
Okay, so let's start there. Section 1, the scout. I usually think of B cells as these factories that just sit in our lymph nodes waiting for orders, but this paper says that's not the whole story.
No, not at all. This study really changes how we view what's called a naive B cell. So these are B cells that haven't fought anything yet. To really see what was going on, the researchers used something they call tomato mice.
Wait, what? Tomato mice, like they're red?
Close. Yeah. They're genetically engineered, so their B cells glow this bright fluorescent red like a tomato under a microscope. It let the researchers track exactly where these cells go in a living animal.
Okay.
And they found something surprising. Naive B cells don't just sit around. They patrol. They are scouts, circulating through tissues, including the lung and, crucially for our story, the brain.
The brain. I thought the brain had a big do not enter sign for immune cells. The blood-brain barrier.
It does, normally. But what the study showed is that during a viral infection, they modeled this using a virus called VSV or even just doing general inflammation. That barrier gets a little leaky.
Like the bouncers on a coffee break.
Exactly. And during that little window, these naive B cells can slip into the brain parenchyma, the actual brain tissue.
So the scout is inside the perimeter. Now what? This is where it gets really visual for me. The paper describes something called antigen capture. What is that?
This is a fascinating, almost violent mechanical process.
Yeah.
So imagine the B cell is sort of like a room of vacuum, just bumping along. It bumps into an oligodendrocyte. That's the cell that makes a myelin sheath.
Okay.
The B cell has receptors on it looking for specific targets. In this case, it's looking for a protein called MOGI.
MOGI.
Myelin oligodendrocyte glycoprotein. It's a protein that sits right on the surface of the myelin. Now, if this patrolling B cell happens to be MOE reactive, meaning its programming is to recognize MOE, it doesn't just touch it. The paper shows it physically tears the protein off the membrane of the brain cell.
Wait, hang on. So the B cell just rips a piece of the brain off?
Literally rips it off. It extracts the antigen. This process is antigen capture. The B cell pulls on the MOGI until it just detaches from the oligodendrocyte. And that is the first step of activation. The B cell is now holding what it thinks is a piece of the enemy.
That sounds incredibly aggressive. I mean, if our bodies are doing this, just ripping pieces off our own brain cells because of a little inflammation, why don't we all have MS? It feels like a massive design flaw.
It would be. It would be if we didn't have a fail-safe. And that brings us to the safety valve. This is the biological checkpoint that, keeps us healthy.
Right, because usually the immune system has a way to stop friendly fire.
Exactly. It's called tolerance. Think of immune activation like a nuclear launch sequence. You need two keys. Key one is the antigen, the B cell finding the threat, or in this case, the myelin.
Got it.
But key two must come from a helper T cell. The T cell has to come along and confirm, yes, that's a threat, you have permission to fire.
What if there's no T cell there to give the high five?
Then the B cell commits suicide.
Wow. That's harsh.
It is, but it's
Yeah.
absolutely necessary. The technical term is activation-induced cell death, or AICD. And the study showed this beautifully. In their mouse models, when these MOG-reactive B cells went into the brain and grabbed the myelin, they just disappeared within days. They died.
What? So the system works.
Under normal circumstances, yes.
Yeah.
1, the B cell enters the brain. 2, it grabs myelin. 3, it looks around for T cell backup. 4, there's no backup because the body knows myelin is self. 5, the B cell initiates self-destruct. And the result, no MS.
Okay, that makes perfect sense. So before we get to how this whole system breaks, let's just do a quick recap to lock this in.
Let's do it.
All right. Bullet 1. We learned that naive B cells are natural scouts. They patrol our bodies, and if there's an infection or some inflammation, they can sneak into the brain.
Once they're inside, they can perform antigen capture, which means physically ripping proteins like MOG right off our brain cells.
And this is the key part. If they do this to our own tissue without a second opinion from a T cell, they undergo AICD. They self-destruct to protect us.
A perfect summary. That is the normal, healthy state of affairs.
So we have this ruthless safety system. How does the disease happen? What breaks the switch?
This is where our villain finally enters the story. The Epstein-Barr virus. Specifically, a protein that EBV makes when it's lying dormant. It's called LMP1, latent membrane protein 1. This protein is the glitch in the system.
How so? What does LMP1 actually do?
Well, remember that key 2, that second signal the B cell is waiting for.
Yeah.
The one that's usually provided by a T cell through a molecule called CD40 ligand.
Right, the permission to fire signal.
Well, LMP1 is a mimic. It looks and acts exactly like a constantly active CD40 signal. Think of it as a fake ID, or maybe better, a hacked key card.
A hacked key card. I like that.
The virus infects the B cell. The B cell goes into the brain, grabs the myelin, normally it would die, but the virus inside it is producing LMP1, and LMP1 just tells the internal machinery of the B cell, hey, we got the signal, we're good to go, survive and multiply.
Even though there's no T cell there at all.
Exactly. The virus completely overrides the safety switch. It tricks the B cell into thinking it has permission to launch an attack.
That is just devious. Did they prove this in the lab?
They did, with a really brilliant experiment. They genetically engineered another set of mice. We can call them the rescue mice. In these mice, they could force the B cells to express this viral LMP1 protein.
And what happened?
When these D cells went into the brain and grabbed the myelin, they did not die. The LMP1 rescued them from AICD.
And not only did they survive, right, the paper mentions a synergistic interaction. What does that mean here?
It means the effect was multiplied. The B cells didn't just survive. When they had both the myelin antigen, so key 1, and the fake LMP1 signal, key 2, they went into overdrive. They proliferated. They started multiplying rapidly, creating this clone army of self-attacking B cells right there inside the brain.
So the virus isn't just keeping them alive, it's turning them into a factory for more soldiers.
Yeah, correct. And this offers a really specific explanation for why it's EBV. Why this one virus is so tightly linked to MS. Other viruses cause inflammation, sure, they might let B cells into the brain, but they don't have LMP1. They don't provide that fake CD40 signal. Only EBV has the specific key to hack the B cell's death switch.
That is the aha moment. It explains the specificity. Okay, so now we have a survivalist B cell army in the brain. They've hacked the system. What happens next? How do we get from surviving cells to the actual lesions, the holes in the brain's wiring that define MS?
Right. This is section 4, the damage.
Yeah.
Once those B cells survive, they differentiate. They turn into plasma cells and they start pumping out antibodies.
The anti-MOG antibody.
Exactly. They're basically tagging the myelin for destruction. And the study details exactly how that destruction happens. It's not the B cells eating the myelin themselves. They call in the airstrikes.
Airstrikes being?
The complement system and microglia. The researchers found deposits of proteins called C1q and C3 all around the lesions. This is part of the complement cascade. It's basically chemical bombs that the immune system uses to blast holes in targets.
And the microglia, those are the brain's own immune cells, right? Like the janitors.
Yeah. Right. But here, they turn into a demolition crew. The study found these dense clusters of IBA1 positive cells, that's microglia, just swarming to tag myelin. And to prove it, they used a drug called PLX5622 on brain slice cultures. This drug, it depletes the microglia.
So what happened when they took the microglia away?
The myelin damage stopped. even with the B cells there, even with the antibodies there, without the microglia to actually eat the tissue, the lesions just didn't form.
That is a terrifying image. The B cell tags it with a laser pointer, and the microglia comes in and just devours it. Let's pause and recap this mechanism before we get into the human connection.
Sure, let's break it down.
Okay, bullet 1. The glitch is the viral protein, LMP1. It acts as a fake survival signal, mimicking what a T cell would normally do.
It allows the myelin-eating B cells to survive that death checkpoint that should kill them.
These survivors then multiply and become antibody factories, tagging the myelin.
The actual damage is done by the complement system and microglia, which attack the tagged myelin, creating the lesions we see in MS.
You've got it. That is the step-by-step mechanism of lesion formation.
So now let's zoom out. We have this perfect mechanism in mice. How does this connect to real people? To the epidemiology of MSS? This brings us to the two-hit hypothesis.
Right, and this is where the paper proposes a model that fits the human data incredibly well. They call it the two-hit model. Hit 1 is the seed. You must be infected with EBV. This creates a pool of B cells in your body that are latently infected and are capable of expressing LMP1.
Which, as we said, is like 95% of adults. So most of us have hit 1.
Correct. But then you need hit 2, the soil. You need an event that opens the door to the brain.
The leaky border.
Exactly. The paper notes that CNS infections or head trauma, especially during adolescence and early adulthood, are major risk factors. If you have a bad case of viral encephalitis or a severe concussion between the ages of 11 and 19, your risk of MS triples.
Triples, that's a huge jump. Why does the age matter so much?
It aligns perfectly with the timeline. You acquire EBV in childhood or your teens, that's hit 1. Then in your late teens, you have some kind of brain event, hit 2, that disrupts the blood-brain barrier. The EBV-infected B cells rush in, they see the myelin, and instead of dying...
LMP1 saves them and the whole cascade starts.
Precisely. And here is a detail from the paper that actually gave me a bit of a chill. They asked a simple question. Do healthy people without MS have these myelin-eating B cells? Yes. They isolated MOG-reactive B cells from the blood of perfectly healthy human donors. We all have the potential for this specific autoimmunity just lurking in our blood.
It's like we all had these sleeper agents.
Right. But in healthy people, those agents either never get into the brain, or if they do, they die via AICD because the LMP1 signal isn't active right at that moment. MS requires that perfect storm, the right cell, the right virus, and the right opportunity.
That's fascinating. The paper also mentioned something a little controversial about finding LMP1 in human brains. Can you touch on that?
Yeah, this has been a debate for years. Some labs find EBV in MS brains, others don't. It's technically very hard to stain for. But this study did stain human MS lesions and found rare but present B cells expressing both CD20, which is a B cell marker, and LMP1.
They're there.
They aren't everywhere, but they are there, which supports the theory that they are the instigators. And clinically, this explains why drugs like rituximab and ocrelizumab work so well.
Those are the B-cell depletion therapies, right?
Yes. For a long time, we didn't fully get why killing B cells stopped MS because MS was thought to be a T-cell disease. But this model explains it perfectly. By wiping out all the B cells, you're removing the scouts. You're removing the only cells that can start the lesion by surviving that death checkpoint.
You're taking away the spark so the fire can't start.
Exactly.
I want to look at the algorithm the paper presents. I mean, they practically gave us a flowchart for how this disease works.
They really did. It's like a decision tree for the cell.
A viral infection or some trauma breaks the blood-brain barrier.
B cell infiltration.
Antigen capture the B cell, grabs the myelin. And now you get to the decision node.
Okay.
If there is NOEBV or LMP1 present, the path is AICD, death, health. But if EBV and LMP1 are present, the path is rescue, proliferation, lesion, MS.
It's binary. It all hinges on that one viral protein.
And we should be rigorous here about the evidence. The paper is very honest about this. The mechanism is proven in the mouse model. That's high-quality experimental causality. The human connection is observational and correlative. It fits the pattern, it explains why the drugs work, but we can't ethically infect humans to prove it directly.
Of course not. But I had one question while reading. We talked about T cells being the backup that the B cell is waiting for, but in MS, aren't T cells also attacking the brain?
That's a great catch. The paper addresses this. They found that eventually these surviving B cells do recruit T cells. In their experiments, they use what are called 2D2 T cells. These are T cells specifically programmed to hate MOG. And they found that the B cells start the process, but once they're established, they present the antigen to the T cells and basically say, hey, come join the fight.
So the B cells are the scouts that light the signal fire, and the T cells are the heavy infantry that come in later to escalate everything.
Yes. T-cell activation was much, much higher when these LMP1 B cells were present. So it starts with B cells, but T cells definitely amplify the damage later on. It unifies the B cell and T cell theories of MS.
That is a massive synthesis. Okay, let's bring all this together for a final recap before we wrap up.
We'll do it.
Bullet 1. MS requires a perfect storm. You need the autoreactive B cells, which we all have, the EBV infection hit 1, and then brain access through trauma or another infection hit 2.
Healthy humans have the ingredients, but we usually lack the trigger. The safety switch, AICD, works for us because the virus isn't active in the right place at the right time.
This model explains why B-cell killing drugs work so well. You kill the hacked scouts. And it provides huge support for developing an EBV vaccine. If we stop the virus, we remove the hacker.
That's the big picture. Absolutely.
This paper really shifts the blame, doesn't it? For years, we looked at the immune system as just being confused. But this suggests the immune system is being sabotaged. The B-cell isn't just a soldier following bad orders. It's a scout that gets hacked by a virus to become a saboteur.
It completely changes the philosophy of the disease. It's not just autoimmunity. It's a viral-driven subversion of tolerance.
So here's my provocative thought to leave you with. Right now, we treat MS with a sledgehammer. We kill all the B cells. It works, but it leaves patients vulnerable. If EBV, and specifically this LMP1 protein, are the drivers, could we treat MS with a scalpel instead? Could we develop a drug that targets just the LMP1 protein? Or find a way to unhack the B cells so they remember how to die when they're supposed to?
That is the billion-dollar question. If you could restore that AICD checkpoint, you could potentially cure the disease without compromising the entire immune system.
And for everyone listening, think about this. If you had a bad concussion as a teenager, or a really terrible bout of the flu that gave you a splitting headache, did that moment open a door for a virus you caught as a child to change your brain's future? It's a little scary, but, understanding the mechanism is always the first step to stopping it.
Absolutely. Knowledge is power.
Thanks for diving deep with us today. We'll see you on the next one.